Hello students. So last class we studied about the digestion of proteins. So in today's class we will study about the absorption of proteins. Now before going into the absorption, let us memorize what we studied in the last class. Okay, that is about the digestion of proteins. So I said cooking and HCl will promote digestion of food. Enzymes involved in protein digestion are secreted as proenzymes and activated in the GIT. Okay, and these will hydrolyze specific bonds at specific optimum pH ranges. Now, I told you these proteolytic enzymes are of two types, endopeptidase and exopeptidase. Now these endopeptidase present in the gastric juice is nothing but pepsin. In the pancreatic juice you have trypsin, chymotrypsin and elastase. What about the brush border of the intestine? You have what is called as enteropeptidase. All of these are basically endopeptidase in nature. The exopeptidase which are present in the GIT, in the pancreatic juice, you have carboxypeptidase which are of two types, carboxypeptidase A and carboxypeptidase B. And in the intestinal brush border, you have aminopeptidase, you have tripeptidase and some amount of dipeptidase. Now, in the cytosol of the enterocytes, you have some amount of tripeptidase and major amount of dipeptidase. So now with the action of all these, the proteins are broken down or digested to the end product called as amino acids with trace amount of tripeptides and dipeptides. Now with this brief recalling of digestion, we will go into the absorption of proteins proper, and at the end of the class you should be able to mention the different mechanism of amino acid absorption, what are the different transport systems of amino acid transport, and what are the disorders associated with defective digestion and absorption of amino acids. Now coming to the absorption of amino acids, students, the site for absorption is small intestine and to some extent in the renal tubules. The mechanism involved is mainly by active transport, it is called as secondary active transport, and to some extent by a process called as gamma glutamine cycle or Miester cycle. Now, the L amino acids are basically absorbed by active transport and they require specific carrier proteins and energy. This transportation is dependent on sodium ion. So it is called as sodium dependent co-transport. In the intestinal lumen, students, you have the amino acid and the sodium. The brush border of the mucosal cell, there are specific transporters, okay, there are specific transporters, and these amino acid transporters have two active sites, and one active site has to be filled by sodium and the other active site is filled by amino acid. Now this energy for this movement from the lumen into the mucosal cell is got because of sodium. So along with sodium, amino acid is also transported into the mucosal cell. So it enters the mucosal cell along with sodium. Now this amino acid from the mucosal cell enters into the interstitial fluid and from there into the portal blood. and then from the portal bud it is taken to the liver where it can synthesize protein. Now, to maintain the electric gradient, when 3 sodium ions are pushed from the mucosal cell into the portal blood, 2 potassium ions are pushed into the mucosal cell using. Now remember, there are 6 different amino acid transporters present for the transportation of different amino acids. This is the first one is called as the transporter for acid, neutral amino acid, and it will transport phenylalanine, tyrosine, tryptophan, methionine, valine, leucine and isoleucine. The second one is the transporter for small neutral amino acids like alanine, serine and threonine. The third one is the transporter for acidic amino acid, like the amino acid transported are glutamate, aspartic acid. Then you have the transporter for basic amino acid like arginine, lysine, ornithine and cysteine. Then you have the fifth type for amino acid like proline, hydroxychloroquine, proline and glycine. Then you have the transporter for beta amino acid, that is beta alanine and taurine. In addition to that, there are transporters for tripeptides and dipeptides also. Now, the amino acids are absorbed passively. So the next mechanism of absorption to a lesser extent which is seen in the intestine, brain and kidney is by a mechanism called as Miester cycle or gamma glutamyl cycle, which operates to transport of amino acid across the cell membrane. Now just see the amino acid on the outer surface of the plasma membrane is pushed into the cytosol by the help of an enzyme called as GTT or GGT, which is nothing but gamma glutamyl transpeptidase. Now this combines with the amino acid in the presence of GGT, combines with glutathione which is present in the cytosol, and it forms what is called as gamma glutamyl amino acid, giving out a dipeptide, that is cysteine glycine. Now this gamma glutamyl amino acid gives out the amino acid, and then you have what is called as 5-oxoproline in the presence of an enzyme, in the presence of an enzyme called as gamma glutamyl cyclotransferase. Now this 5-oxoproline gets converted to glutamate in the presence of oxoprolinase. This requires a molecule of ATP, that is ATP is converted to ADP. Now if you see, this dipeptide cysteine glycine undergoes hydrolysis in the presence of peptidase to the amino acid glycine and cysteine. Now this glutamate later combines with cysteine to give gamma glutamine cysteine in the presence of synthetase using ATP. Now later this gamma glutamine cysteine combines with glycine using ATP and forming glutathione in the presence of an enzyme called as glutathione synthetase. So when a molecule of amino acid crosses the plasma membrane, 3 molecules of ATP are used up. Is that clear? So one here, now one here. Next result is the transfer of amino acid across the membrane. But remember, they cannot transport amino acid. So 3 molecules ATP consumed, 1 molecule of amino acid is transported across the membrane. Very small amount of intact proteins are able to cross the intestinal epithelium by a process called as pinocytosis, especially in the infants. Here the immunoglobulins, this is seen in infants. Here the immunoglobulins secreted into mother's milk, that is colostrum, are absorbed by the intestine, by the infants, which gets immunity until it begins to produce its own antibodies. But remember, when other proteins are absorbed, it may give rise to immune reaction. That is what we see in food allergy. Example for this is allergy to egg protein, milk proteins and other food proteins. So that is all about absorption. So absorption basically takes place by active transport and to some extent in the intestine, brain and the renal tubule by gamma glutamyl cycle or Miester cycle. Now coming to the disorders associated with digestion and absorption of protein. First one is enteropeptidase deficiency. If you remember the first class, I said enteropeptidase is required for the activation of trypsinogen to trypsin, and in turn trypsin activates other enzyme, proteolytic enzyme like chymotrypsinogen, proelastase, procarboxypeptidase, etc. So now what happens is, when this enzyme is deficit, activation of trypsinogen is decreased and therefore other proteolytic enzyme activity is also decreased. So this results in impaired protein digestion and absorption and also growth failure. Now as a result, the plasma protein level is decreased and there will be, the patient will be edematous. Okay, that is one type. The second type is called as gluten sensitive enteropathy. Now here this is a malabsorption observed on ingestion of wheat grains, gluten in wheat protein gluten in genetically susceptible individual. This gluten injures this causes injury to the intestinal mucosal cell, and because of this, the microvilli or the villus of the brush border gets atrophied, okay, and it will result in absence or reduction in the height of the villi. So this is a normal villi and here it is the damaged villi. You can see here this is a normal villi and here the shortened damaged villi. Now because of this, what happens, there is malabsorption and consequent symptoms like diarrhea, steatorrhea, weight loss, abdominal pain and fatigue, and symptoms of nutritional deficiency like anemia and bone abnormalities. Also there will be, this is more common in Caucasians, and if you avoid food like wheat products, barley, rye, oat, etc., which does not contain gluten, which contains gluten, may relieve the symptoms. Next disorder is Hartnupp's disease. Here there is a defect in the renal and intestinal transport system of neutral amino acid. So there is decreased absorption of these neutral in the there's decreased absorption of neutral amino acid in the GIT, and that there will be increased excretion in the urine. And all the manifestation of Hartnupp's disease is due to decreased tryptophan level. Tryptophan is a neutral amino acid. So there will be decreased niacin production. As a result, there will be pellagra-like symptoms and skin rashes, and there will be intermittent cerebral ataxia. This is a patient suffering from Hartnupp's disorder. Yes, see the rashes, skin rashes, and you can see the Castle's necklace, the skin rashes called as Castle's necklace in pellagra. Mental retardation, psychiatric symptoms could be seen, and this gets exaggerated in fever, stress and stress condition. So there will be increased excretion of indican, indole 3 acetic acid and other degradation product due to microbial metabolism of this unabsorbed tryptophan. The other condition is called as cystinuria. Here there is defective system of absorption of basic amino acid and cysteine. So cysteine will be excreted in the urine. Dicarboxylic amino aciduria, there will be defective transporter for dicarboxylic acid. So it won't be absorbed and excreted in the urine. So that is all about digestion and absorption of protein. Now, I want to emphasize one more point, clinical point, that is acute pancreatitis. Now, in acute pancreatitis, student, there is premature activation of trypsinogen inside the pancreas itself, and because of this, there is auto-digestion of the pancreatic cell, and this condition is called as acute pancreatitis, which is a life-threatening condition. So, with this, the whole story is over. Once again, like a chewing gum, I repeat, cooking and HCl promote digestion of food. Enzymes involved are secreted as proenzymes and activated in the GIT. They hydrolyze specific peptide bond at specific optimum pH. These proteolytic enzymes are endopeptidase and exopeptidase. I have enlisted all this. In the mucosal cell, you have tripeptidase and dipeptidase as well. So the end product of protein digestion is amino acid, and these amino acids are absorbed by specific transport systems and also to some extent by gamma glutamate cycle. So what are the disorders? One is enteropeptidase deficiency, then you have celiac disease or gluten sensitive enteropathy, heart nerve disease, cystinuria, dicarboxylic amino aciduria. That's all about digestion and absorption of proteins. Okay. Thank you for listening to my class, students, and I would be sending you a small quiz on digestion and absorption. Please attempt that, and also if you have any doubt regarding this topic, you can approach me at any time. Thank you.